So can I start? VC sir, can I start?
Yes, madam.
Okay. Thank you, sir. Hello, everyone, and welcome to today's international webinar entitled Zebrafish, an emerging model in research. I, Dr. Himanshu Gupta from Department of Medical Biotechnology, MGM School of Biomedical Sciences, Navi Mumbai, take this opportunity to acknowledge our patrons, Honorable Vice Chancellor, MGM IHS, Dr. Shashank Dalvi, Honorable Pro Vice Chancellor, MGM IHS, Dr. Nitin Kadam, Registrar, MGM IHS, Dr. Rajesh Goel, Dean, MGM Medical College, Dr. Narishetty, and Director, MGM School of Biomedical Sciences, Dr. Mansi Thakur. I also welcome our acclaimed speakers, Dr. Anirban Chakrabarti, Dr. Chetna Sachidanandam, and Dr. Patrick Sips, who will enlighten us with their talk. A warm welcome to all the delegates who have joined in from India and abroad as well. The most interesting thing about zebrafish is their ability to regenerate. That is, when they lose something, an eye, a fin, a tail, even individual cell types, they grow a new one. In addition to its many advantages, their genetic profile is remarkable, similar to our very own, whereby they provide scientists with genetic clues to medical mysteries. I myself have worked on zebrafish for my PhD, where we studied on the mechanism of action of homeopathic drugs on GIOP model, that is glucocorticoid induced osteoporosis model on zebrafish, which we have standardized in our laboratory. It can be said that while science has focused on mice and rats as models to study diseases, since the creation of the first genetically altered mouse in 1980, zebrafish have in recent years emerged as an important model species as well. I consider it my great honor to request Honorable Vice Chancellor, sir, to address the audience. Over to you, sir.
Thank you, Himanshu. Now, as a Vice Chancellor of MGM Institute of Health Sciences, Navi Mumbai, I would like to welcome all the faculty and the participants, the students who have joined for this particular webinar. Department of Biomedical Sciences have selected a very relevant topic, the zebrafish upcoming research model. Now, it is not available at all places, or everybody is not very much aware about this, but this is very convenient, useful model that has been upcoming. And also regarding the animal experiments, the various legal bindings are there. So for this particular model, as such, those legal bindings are not that much constraining, and so this particular model is very, very useful. And I actually congratulate the whole biomedical science department, the staff, the in charge, Dr. Mansi, for this particular enthusiasm and organizing this particular international webinar, where the faculty from different parts of the country, like Dr. Anirban Chakrabarti, Dr. Chetna Sachidanand, and Dr. Patrick Sips, is guiding you, informing you in this particular webinar. And I am sure this webinar will give the idea about this particular zebrafish upcoming research model, and more scientists will turn to use of this particular model. I wish all the success to this webinar. Thank you for inviting me to this particular, giving the address for this particular webinar. Thank you very much. Thank you very much.
Thank you, sir. Thank you, sir, for your kind words of encouragement. With equal pleasure, I now request Director, MGM School of Biomedical Sciences, Dr. Mansi Thakur, to say a few words. Ma'am, please.
Honorable Vice Chancellor, Dr. Shashank Dalvi sir, Honorable Pro Vice Chancellor, Dr. Nitin Kadam sir, Registrar, Dr. Rajesh Goel, and Dean Medical College. I am really grateful to the speaker who has accepted our invitation to be a part of this international webinar. Dr. Anirban Chakravarti, Dr. Chetna, and Dr. Petri. I am really happy to share all of you who are the delegates presently in our webinar. More than 400 registrations we got, not only from the India, but from the other part of the country. As we all know that main delegates are our students, and we have focused this zebrafish model for our students only. So I want to share a few things about the student as a delegate. We all know that we have many model organisms whom we are going to study. Like we all know that the bacteria, fungi, viruses, prokaryote, plant, animals, many are the models which are under studies from last many years. But in vertebrate, also we have many models like cat, mouse, and recently zebrafish come up in the last few decades. Zebrafish come up as a extensively used vertebrate animal model. This model is used for especially for the studying the development, toxicity, and neurobiology. We have specifically planned this webinar to exhibit the modest zebrafish as a model for research to the young generation. Why zebrafish? Because zebrafish has a unique advantage for understanding the evaluation of vertebrate immunity and to model human disease. With the innovation and improvement of molecular techniques, zebrafish will continue to be an important biomedical model in future. So I am not taking much of time. I request Dr. Himanshu Gupta to take ahead the session, and all the very best to Department of Medical Biotechnology as well as all the delegates who are present in the session. And I hope everyone will get the insight how this emerging model is useful for your studies in future also. So wish you all the best and thank you for all your participation.
Thank you, ma'am, for your never-ending support and encouragement. You're an inspiration for us. Without further, without further ado, I'd like to introduce the first speaker, Dr. Anirban Chakrabarti. Sir is a professor and deputy director at NITI University Center for Science Education and Research, Mangalore. Sir is the founder for Consortium of Rare Genetic and Bone Marrow Disorders, India Network at NITI. Sir is a doctorate in agricultural science and postdoctoral fellow from University of Miyazaki, Japan. Sir has various awards to his credits, like the Best Researcher of NITI, earlier Career Research Award by Science and Engineering Board, DST, MEXT Fellowship by Japan, Government for PhD. Sir is also a PI in various government-funded projects. Sir has over 50 publications in national and international journals with cumulative index factor of 110.91. Sir's research interests are ribosome and human diseases, cancer biology and genetics, nuclear proteins and their role in tumorigenesis and cancer, zebrafish developmental biology, small molecules, drug screening. Sir is going to talk on modeling rare genetic disorders in zebrafish. Over to you, sir.
Thank you, Dr. Himanshu. I hope I'm audible to all of you. Thank you, Dr. Mansi. First of all, it's nice to get connected again after 2017. I guess that was the time when Dr. Mansi was here in NITE, and then Dr. Himanshu also participated in the next conference as well. So it's a great feeling to get connected. And since Dr. Mansi mentioned that majority of the participants are students, so what I'll do is I will take you through this journey of the zebrafish in my research career, and I will, 1 second, I have this issue here. Okay. Yes. All right. Okay. So let me open my presentation. I hope my presentation is visible.
Yes, sir. It's visible, sir.
Okay. So let me begin with, I have, Dr. Himanshu told me I have 45 minutes to speak. So I will try to go in a very simple manner. It's ideally to enthusiast the students to know about what all we can do with the zebrafish. So I work with this model system, and I basically focus on ribosomal diseases, which are rare genetic conditions in human. So basically, I think my speakers who are going to speak later, they're also going to introduce this model system to all of you. But maybe because I get this privilege to be the first one to speak, so let me start with a bit of introduction. I'm sure most of you or many of you would know what this model system is, how this name came up from, and what are the applications of this model system. So basically, zebrafish, it is called because of these horizontal stripes that they have their body, belongs to this family Cyprinidae, which is the largest family of fish in the kingdom. So in simple terms, I think they belong to the group of the carps, which are very common in my part of the country. The good part about this model system is, in addition to its several inherent advantages, I think one of the most important feature is its ability to regenerate its skin, fins, and heart, and to an extent, kidney as well. Ideally, the model system was developed for studying embryonic development or developmental biology, or it was proposed to be an ideal model for studying early development. But then people quickly realized that this model system has a lot of other applications. And in today's context, I think there is hardly any disease or any condition, human condition, that has not been modeled in zebrafish. So tremendous applications, and I think the Honorable Vice Chancellor was mentioning that there are certain advantages as far as the ethics are concerned. Yes, we do have certain advantages. It is not yet under the purview of the animal ethics, but very soon, zebrafish is also going to come as one of the organisms where you need to have an ethical clearance to conduct research on it. So the ability of this organism to become a very important model is their extremely rapid development and the ex utero development that happens outside the body. So it grows about 4 to 5 centimeter. That's the maximum size. Survives for about 4 to 5 years, but in experimental conditions, we don't use them beyond 2 years. And originally from India and the neighboring countries, but somewhere somebody in Germany actually realized the importance of this fish and started with the experiments on developmental biology. Since then, it has been a very satisfying journey for everyone involved in zebrafish research, and also those who are yet to get into this. I'm sure in the next 40 minutes, I will try to convince you that this is a model that one can really, really look into. I have a small video for you here, just to tell you about how the zebrafish develops. So this is actually, the video captured from one cell to the stage where it becomes fully hatched larvae looking for food. So as you see here, the cell division is taking place, and then you have this shield stage coming up, and then by about 8 to 10 HPF, you would probably look at the sulfate, which are going to, which is making this.
So sorry to interrupt you. Your slides are not moving ahead, sir. We are still on the title slide.
Slides are not moving ahead?
No, sir. We are still on the title slide.
I don't know the reason. Okay, 1 second. Just
yes, sir.
Okay.
Yeah, now it is.
Okay, is it moving now?
Yes, sir. Okay. We are on the, yeah.
So, all right. So what I was talking about was this video that I'm showing you here. Can you see the video, Dr. Himanshu?
Yes, sir, I can see.
All right. So, so basically, everything that is happening in this embryo, fertilized embryo that is growing, I think you can see them from outside. All you need is a good quality stereo microscope and a good time-lapse video camera, which can capture the fascinating changes that are happening in this embryo. So I was mentioning that by about 8 to 10 HPF, it actually takes the shape of an animal, and then you already can make out the somites are forming here, and then by 18 HPF, you would have almost miniature embryo coming up, and by 24 to 25 HPF, you have fully developed brain, and by 48 hours, you have the development of the vasculogenesis complete. You can see the pigments started coming up by day 2 or by 48, and then the embryo is taking a full shape, and the mouth comes up by day 2 or day 3, and then they hatch out of their chorion, and then they start looking for food. So that is the free swimming larvae, or the completely hatched larvae that comes out by day 3 or day 5. As you see here, the gut is also forming, the gut associated organs are coming up. So it's very nice to see that the animal is fully grown from one such stage. All of these happen in just 3 days' time. To put things into perspective, human embryo takes 24 hours to divide once. So zebrafish is almost a miniature adult in 24 hours. So quite fascinating, quite rapid as far as the developments are concerned. So advantages, I think we all know that there are so many inherent advantages. Probably the most important one would be the optical transparency of the embryos. You can actually see the development, all the organ development right from outside of the body. You can have extremely rapid development. Within 3 months, you have a complete adult, which is ready to mate. And if you're talking about transgenesis, or you're talking about developing mutants, I think that is a very good time frame to have a mutant within the F2 generation. You can have within 6 to 8 months, you have an F2 generation. High fecundity, it gives about, one female gives about 100 to 200 eggs in a week, which is a very good number compared to your higher mammalian model systems. So you can have an experiment with high levels of statistical significance and high levels of reproducibility. Genetic information, I think the biggest advantage why zebrafish took or got the attention all over the world is perhaps its extensive genetic information and the similarities that it has with human. So if you look at the protein coding genes, about 75% homology you have, and particularly if you're talking about cancer-related genes, or in the genes that are responsible in the cancer pathway, you have more than 80-85% similarities. The kind of genes that I work with, ribosomal protein genes, or the genes that are involved in ribosome biogenesis, they have more than 90 to 95% similarity with the human. So quite a useful model. You have genetic manipulation tools, plenty of them in zebrafish, both forward and reverse genetics. So overall, it's a good package to have such a model system in your research lab. And of course, one could say that, yes, it is not a mammalian system. But yes, it is not a mammalian system, but it's a vertebrate system, which shares a lot of similarities, particularly in the field of cancer biology. So major applications one can think of is in toxicology, in pharmacology, in functional genomics, and disease modeling. So today's talk is about disease modeling. And again, under that, it's about a rare genetic disorder that we're talking about. So I told you in the beginning that I work with ribosomes. So before we get into the modeling part, let me give you a brief introduction about ribosomes, what they are. Of course, all of us know it is the most complex macromolecule that is in a cell. Ideally, the protein synthesis factory that we talk about. What we don't know much is that the process of ribosome synthesis is a very complex and well-coordinated process. It starts at an organ called nucleolus, which is an organless or membraneless organelle, which is formed within the nucleus. And then the maturation of ribosome happens throughout the cell. And finally, the final assembly of the subunits happen in the cell, in the cytoplasm of the cell. So in an eukaryotic system, we have 79 ribosomal proteins and 4 ribosomal RNAs. Together, they constitute the mature ribosome. And I already mentioned that these ribosomal proteins and ribosomal RNAs are highly conserved right from yeast to human. So it is very easy to work with these genes. And if you're talking about modeling them, it becomes even more easy affair to talk about their function across evolution. So what happened in the field of ribosome research or ribosome biogenesis, the landmark year was 1999. So something was before and something happened after. So till 1999, the concept was that ribosomes are extremely critical for cell survival, and one cannot survive with defective ribosomes because we know ribosomes are ubiquitous. So you cannot imagine a cell without a functional ribosome. In 1999, a paper came out in Nature Genetics where the researchers identified mutation in a ribosomal protein gene called S19. So this is a gene which codes for a ribosomal protein, and that protein plays a role in the 40S subunit synthesis. And what they found is this gene is mutated in a rare genetic condition called Diamond-Blackfan anemia. I will talk about this as I go on with my presentation. So this disease was first reported in 1966, 1967, if I'm correct. But it took 30 odd years to identify the causative gene. That is because nobody would have imagined that the mutation could be in a gene which is so important, so very critical for cell survival. Once this was found, and they found that people, so they are, they're not dying because of this mutation, although they realized that the ribosomes are defective in these patients. So after 99, the notion changed. So what happened? So now we know that faulty ribosomes does not make a cell inviable, and defective ribosomes cause diseases in humans. So then came the concept of ribosomopathies. So ribosomopathies are basically diseases that are caused due to defects in ribosome biogenesis. In other words, the defects are caused by mutations in the genes that are responsible for ribosome biogenesis. So as I mentioned earlier, in addition to the ribosomal proteins and ribosomal RNAs, there are many associated factors which play a role in ribosome synthesis. So they are called as ribosome associated proteins. So after the discovery of Diamond-Blackfan anemia, or the discovery of the mutation in the causative gene, people have found several other conditions where the causative gene was somewhat linked to ribosome biogenesis. So all these diseases were grouped together and they were called ribosomopathies. So I have made a mention in my slide. These are mysterious and curious. So as I go on, I'll try to explain what is the mystery behind this and why they are curious. So the collection of these rare genetic conditions where the causative gene is either a ribosomal protein gene or a ribosome associated gene, they are called ribosomopathies. And the most intriguing part of this group of diseases is that they produce tissue-specific phenotype. In case of Diamond-Blackfan anemia that I work with, the phenotype is in the hematopoietic system. So again, the mystery starts right there. Ribosomes are required for all cells. Why is it that the patients with ribosomal defects or defects in ribosomal proteins have defects only in their hematopoietic system? Why not other organs? So this is something which has not been known, which people are still trying to understand, but the answer is still not clear. Why is it so? But the concept of specialized ribosomes has come up where people are saying that some ribosomes are required for some organs, and certain ribosomal proteins are indispensable for those organs. Anyway, so this is the beginning of this. So Diamond-Blackfan anemia, as I said, it's a classical example of ribosomopathy. If you consider all the ribosomopathies that have been identified so far, the most extensively studied is Diamond-Blackfan anemia. Mainly because it is more common, and there are a lot of repositories and foundations around the world who are working on this. And I've been fortunate to be a part of the French registry, but there are registry in Japan, in France, in UK, in all the European countries, more or less, they have a registry of their own. We have started something since 2019, where we have set up a consortium on rare genetic disorders. Our focus is on these ribosomopathies, and we hope in the coming years we'll be able to set up a registry of our own in India. At the moment, we don't have much of information on these Diamond-Blackfan anemia in the country. So it is a rare genetic disorder, 5 to 10 cases in a million live births. So it is quite rare. So today's talk is about the dynamics and disorders of ribosome biogenesis using the Zebrafish model system. I will take the example of Diamond-Blackfan anemia. We have been working on many other diseases using the same Zebrafish model system, but due to time constraint, of course, we can't talk about all of them. I'll take this example of ribosomopathy, the DBA, and I will take you through this journey of the research that has been happening, or the part of the research that I have been associated with so far. So DBA, a little bit on DBA, it is an inherited bone marrow failure syndrome, about 5 to 10 cases per million live births, as I mentioned already. The good part is that more than 90% of the patients are diagnosed in the first year, so it is a pediatric condition, and it shows up quite clearly. Now, the most common, or the tissue-specific, or the prominent phenotype is the erythroblastopenia. In simple terms, it is less number of mature RBCs in a peripheral blood. The second important part is that they have a normal marrow cellularity. So all the other lineages of the hematopoietic differentiation, all the other lineages are normal in these patients. Only there is a selective deficiency of the red blood cell synthesis, or the red blood cell precursors. Some of the patients also show certain anomalies, pleiotropic anomalies, for example, stunted growth, skeletal abnormalities, polydactyly of the finger, abnormalities of the upper limb, cleft lip and palate, which is also quite strongly related to a particular mutation that have been identified. And then some of them also have kidney dysfunction. Not all, but about 40 to 50% of the patients have cancer predisposition. So they develop all kinds of solid cancers, including hematopoietic malignancies. A little bit of genetics. As I mentioned, RPS19 is the first one that was identified and is the most frequently mutated RP gene that has been known so far. But overall, there have been 18 ribosomal protein genes at the moment identified. I have mentioned only the top 3 groups. The second category is about 5 to 7%. RPL5, RPL11, and RPS10 belong to that. And there are some more which are written over here. In addition to the RP genes, recently people have also found that 2 X-linked genes, that is GATA1 and TSR2, these are also mutated in DBA. Good. We have a fairly good idea about the genetics, but what is not known is what happens to the remaining 35% of the cases. So there are plenty of genes that could be potentially mutated in DBA. People are looking at systematic analysis at all the ribosomal proteins. They have looked into it, but there is so much that can be done. And it is quite possible that mutations could be in any of these ribosome-associated genes. So the question which comes to everybody's mind, it is a disease of the human. Why you don't do the research on mice? Why zebrafish is a preferred system for DBA research? So in mice, yes, we have mice models of DBA. They were the first models to be developed. But then when you look at the RP heterozygous mice, they don't show any hematopoietic defect, unlike the human counterparts. In the patients, it is a heterozygous condition, so they show the phenotype. But if you try to model it in a mice, the heterozygous condition does not lead to any hematopoietic defect. They are absolutely normal. Then the question comes whether you can work with the null mice or the homozygous deleted mice. But then, in case of the homozygous deletion, they are lethal before birth. So they are dead even before they are born. So obviously, you can't work on that. So in case of zebrafish, what happens is, yes, in zebrafish also, heterozygous condition is normal. So there is no hematopoietic defects or any other defects in the heterozygous mice. But the homozygous ones are lethal, but they survive up to 5 or 7 days post-fertilization. And that is a long window to study hematopoiesis. So they are able to survive up to a stage which is well beyond the onset of hematopoiesis. So that makes it a good model or a favorable model to study DBA or any other ribosome-associated diseases. And of course, the RP deficient zebrafish, they are severely anemic. Of course, when you're talking about hematopoietic malignancy, you need to look at whether your model system is able to have the primary phenotype, which is anemia. That is the first thing that one needs to do. So this is my research milestones. So I started with this from 2006, and along the way, we have identified a few things. We have clarified a few discrepancies. We have understood a little bit more on the mechanisms of DBA, and we have complemented to the studies that were going on in cell lines and in mice models. So today's story is about the results that we have obtained from these 6 publications that I have listed out here. And I'll go in the form of a story. We'll start with the beginning, and then I'll try to complete it without too much of technicalities. As I understand, this is mainly a student crowd, so we'll have to keep it simple. All right, so the first thing when you start, you have to do is you have to create a model. So when you create a model, you have 2 ways to go about it. Either you have a genetically inactive system, or you have a system where you can transiently knock down your gene of interest. In zebrafish, we use morpholino for that. So we used a combination of morpholino-induced knockdown zebrafish as well as a genetic mutant, genetically inactivated RP mutant. So the first goal for us was to create an RP deficient zebrafish that recapitulates DBA phenotype. As I said, the most prominent phenotype is anemia. So the first thing that one has to do is to check whether you can do hemoglobin staining or whether you can track the blood cells in the fish. So there is this staining called ODA staining, which is a simple, very simple staining. It takes about 15 to 20 minutes to do it. And this can very clearly and very easily identify all the blood cells. So in 2008, we started with this, and this is the first paper that came up on a zebrafish model of DBA, where we showed that if you knock down RPS19, you can get a severely anemic zebrafish. The images which are shown on top, that is a wild type, which is normal zebrafish. The one bottom is the RPS19 knockdown zebrafish. If you see, as you see here, the brown spots that you see on the belly of the fish, these are the red blood cells, which are which is the common cardial vein, which is captured here. If you see the one in the RPS19, they are absolutely clean. So there is no severe reduction of red blood cells. Yes, good. So we identified this. We found, okay, so RPS19 knockdown fish is a good way to start. So first, when you do that, next is to check whether this defect is only in the erythropoid lineage or whether the defect is on other lineages. If you remember, I mentioned that the patients show a defect only in the erythroid precursors. They don't have any other problem with, say, myeloid precursor or the vascular precursors. So this is a snapshot of how the hematopoiesis happens in zebrafish. So the part which is encircled is a transcription factor called GATA1, which is used for checking the levels of erythroid progenitors or the precursors in the animal. So we looked at this and we found that the RPS19 mutant or the RPS19 morphant had defects with the GATA1 expression, whereas in the myeloid lineage and the vascular lineage, they didn't have such obvious defects. So it looked like they have erythroid lineage specific defects. So good. So we could clear that, yes, they were anemic and that anemia was due to a defect in the erythroid maturation. The second part was to check whether the mutation that were identified in human beings, whether these mutations are pathogenic, and if yes, whether they are able to rescue the defects or not. So we made different forms of RPS19 externally by in vitro transcription. So we introduced missense and nonsense mutation in the RPS19 mRNA. These are patient type mutations, and we also made modified RPS19, which is similar to the wild type RPS19, but only difference is that they are resistant to the morpholino. So morpholino will not be able to bind to this mRNA. So we injected our fish with this, and this is how it looks like. I'll show you the first part of the image. Here you see the injection were done with the modified mRNA, which is the wild type RPS19 mRNA, and they were injected along with RPS19 morpholino. So the morpholino will knock down the endogenous RPS19 mRNA, whereas the modified mRNA will be available for the fish to be synthesized into RPS19 protein. So as you see here, we see a rescue of the anemia. So the brown spots which are missing in the morpholino injected ones, they are coming back in the injected animal which were with the modified mRNA. But if you see in the bottom figure, when we injected them with the missense mRNA and the nonsense mRNA, we don't get back this brown signal, or that hemoglobin staining does not come back. So they do not become non-anemic. They continue to be anemic. So what it means is that you can have an erythroid rescue by RPS19 mRNA, but when you inject DBA type patient mRNA, you don't get that rescue. So this completes the story that yes, zebrafish is a good model. It can be a viable model to study other things related to DBA. So next, we also looked at why this anemia is happening. So basically, we collected the blood cells and we identified that the maturation defect, and then we also looked at that they were dying, or they were apoptotic cells. So the ones which are immature, they were dying because of apoptosis. So we concluded that we developed a zebrafish model, and we found that it recapitulated all the phenotype that we wanted to look at. And we also showed that RBC maturation and subsequent apoptosis were the major contributing factors to the anemia that was visible in this animal. So that was the first part. And then when we started this, then we found that this is the beginning of the mystery. So while we were working on these the model in zebrafish, people were also simultaneously working on human cells and in vitro cells, and they proposed a mechanism for pathophysiology of DBA, and that mechanism was ribosomal stress. So basically, if ribosome synthesis is defective for some reason, this stress is sensed by P53. P53, as you know, the guardian of the genome. So P53 gets activated, and it gets uncoupled from its negative regulator called MDM2, which is shown here. And P53 becomes active, and then it leads to cell cycle arrest or apoptosis. So it does not allow the defective cells to proliferate. And this is where it also takes part in sensing the ribosomal stress, and in a way, it takes part in a surveillance system, which cells possess. So what people found is that, yes, there are many ribosomal proteins involved in it, but then two ribosomal proteins, that is L11 and L5, were the major players. Their participation was extremely critical for this activation of P53. So we, of course, when we developed our model, we wanted to see whether P53 is really involved, and we found that yes, P53 was really involved. So I'm just focusing on two parts. The part which is circled here, this is a RPS19 morphant. You see that head and eye is very small and they have morphological defects. And if you look at the tunnel assay, which is assay to check blood cell, apoptotic cells, you see extensive apoptosis in the animal. But when you remove RPS19 and P53 together, you see a rescue of this phenotype. The embryos become almost like wild type, or almost like the animals where RPS19 mRNA is injected. And you also see there is considerable decrease in the number of apoptotic cells. So what it means that when P53 was active, it was leading to these morphological defects because of apoptosis. So when you remove P53, you don't see these apoptotic defects, or you don't see the morphological defects. So P53 is involved in the RPS19 associated morphological defects. But then a group in the US, Gazda's group, they identified mutation in L5 and L11 in DBA. So this was totally unexpected and totally contradictory to the mechanism that was proposed, because they said L5 and L11 are important. And that is why it is leading to the P53 activation. But then they found that both these genes are actually mutated in DBA. So there must be something else which is happening. So that led us to look at what is the relevance of P53, or what happens to P53 when we remove L11 in zebrafish, whether P53 remains active or it becomes inactive. Actually, we expected that P53 would become inactive, because we were following the pathophysiology which was proposed based on cell line work and based on the work done on patients. So we removed L11, and then we found that L11 actually increases P53 in zebrafish, contrary to the results that were shown in cell lines. And as we have seen for S19, when you remove both P53 and L11, we see a phenotypic rescue. So that led to more questions. So the observation was that TP53 or the P53 is involved in the morphological phenotype. And the question that came up is, yes, it is, but then what happens to the erythroid phenotype, or the phenotype that is responsible in hematopoiesis? So we did a lot of experiments with S19 and L11 deficient fish. These were published separately, but I'm putting them together in a single slide. So what we found that when we removed P53, we got a complete morphological rescue, but we did not get an erythroid rescue. So that means P53 is involved in morphological defects, but P53 is not responsible for the anemia phenotype that we see in the model system. So this is how it shows. If you see here, RPS19 morpholino and RPS19 morpholino plus RPS19 mRNA, you get a rescue of the brown spots. But if you see RPS19 plus P53 morpholino, you don't see the rescue spots as compared to a wild type. Same thing on the L11 condition. So here we looked at different combinations. We took a P53 mutant, we removed L11, we did not find a rescue. Then we took L11 mutant and we knocked down P53. There also we did not find a rescue. And then we took a wild type fish and we knocked down both L11 and P53. There also we did not find a rescue. So it was absolutely clear that P53 is responsible for the morphological phenotype, but not for the erythroid phenotype. Yes. So the next question again came up that yes, it was convincing beyond doubt that P53 is not responsible for the erythroid phenotype. So next question that came up is whether it is true only for DBA type of RPs or any other RPs or any other ribosomal protein. So again, we went on to look at all kinds of ribosomal proteins, regardless of whether they are involved in DBA or not. And we found that in all of them, P53 is not involved in the erythroid phenotype. So P53 knockdown, either in a mutant condition, you knock down the ribosomal protein, or in a wild type condition, you knock down both P53 and ribosomal protein, you don't see the rescue of the erythroid phenotype. So what we understood was that deficiency of any ribosomal protein, regardless of their role in DBA, leads to anemia, leads to erythroid defects, but that is not rescued by depletion of P53. So the next question came was, so what was that causing the anemia in RP deficient zebrafish? Morphological part, we were clear that it was P53, but we are still not able to answer why the anemia was happening in ribosomal protein deficient zebrafish. So then we looked at another aspect. So that was the translational defect. And that was kind of expected because ribosome is involved in a protein synthesis. So if there is a ribosome biogenesis defect, you would have translational defect. So we looked at two amino acids, L-leucine and L-arginine, whether they are known to involve in the mTOR pathway. And as we know, mTOR pathway is involved in enhanced translation. So we found that when we put the RP deficient animal and if we treat them with L-leucine or L-arginine, we not only get a phenotypic rescue, but we also get an erythroid rescue. So the answer was P53 involved in morphological type, but not in erythroid type. So it is the ribosomal defect which was responsible for the erythroid defect, whereas P53 is responsible for the morphological defect. So the observation was that activation of mTOR, which enhanced translation, resulted in the rescue of both the phenotypes. And of course, the knockdown of RP resulted in decrease of global translation, and that led to the consequences. So far, all good. We have found out answers to almost everything that we were looking at. So if the question is, are all mysteries solved? The answer is no. We still had to find out why there was an increased TP53 activity in L11 deficient zebrafish. Because if you remember, according to the pathophysiology, L11 is the most important ribosomal protein that allows P53 to sense nuclear stress. So we still had to find an answer to this, and that led to this. The most recent work that we did was on looking at the c-Myc target nuclear proteins. So c-Myc is a transcription factor, as we all know. It has thousands of targets, but there are some targets which are known to play a role in ribosome biogenesis, mainly in processing, ribosome assembly, and nuclear export of ribosomal subunits. So we looked at, interestingly, these are also responsible for modulating the P53 activity or for modulating the P53 MDM2 axis. MDM2 is a negative regulator of P53. So for P53 to become active, MDM2 has to be taken care of. So these three proteins that I mentioned down, they bind to MDM2 and uncouple these P53 MDM2 axis and allows P53 to become active. On the other hand, MIB is one that modifies P53 and that allows it to become active. So we wanted to look at the role, the expression of c-Myc. c-Myc was overexpressed in L11 mutant, and both by in situ hybridization and by real-time PCR, it showed increased expression. So when c-Myc was increased, so you also looked at the expression of the c-Myc targets. And expectedly, these targets are also overexpressed. We looked at many targets, and we looked at the ones that are responsible for ribosome biogenesis, and almost all of them showed overexpression in L11 mutant. Interestingly, those that bind to MDM2 and modify P53, they are also overexpressed. Then we wanted to look at where these targets are expressed, which part of the animal, and we found that they are overexpressed in the region, the head and eye region, and that is the part which was showing morphological defects in ribosomal protein deficient zebrafish, and P53 also expresses in this part of the animal. So there was a good correlation with the expression of the nucleolar proteins and with the P53. We also went on to do a time point analysis, and we wanted to see at what stage the expression goes high, and we did at different time points, and we found again a correlation between the P53 targets and the nucleolar protein or the c-MYC target nucleolar proteins. So the conclusion to that was that L11 allows increased P53 activity, but at the same time it increased c-MYC activity, and increased c-MYC results increased expression of nucleolar protein, and these nucleolar proteins, which are P53 modulatory in nature, they bring about the activation of P53 in the L11 deficient zebrafish. And this is independent of the MDM2 RP interaction. So conclusion part. So the story so far is that we have ribosomopathies, they lead to defective ribosomes, and two things happen when there is a defective ribosome. One is the nuclear stress, the other one is anomalies in translation. So nuclear stress activates TP53, and that leads to morphological defects. And we have already explained that if you play around with P53, you are able to rescue these morphological defects. On the other hand, defective translation leads to P53 independent anemia, which is the condition where P53 cannot rescue that anemic condition. And that is due to defect in global translation, and that leads to the erythroblastopenia or the anemia phenotype that you see. So what are the evidence that we have? Activation of P53. Again, it is due to increased c-MYC activity in the L11 animal, and this evidence is present from DBA fish model, and then also the anomalous part that is also evident from the DBA fish models. So are we clear now with all the questions? The answer is no. There are many questions which remain unanswered. So some of them are listed here. If ribosomes are ubiquitous, why hematopoiesis is specifically affected, which is something people started with, but so far, so many years of research, but still people are unable to answer this. And if erythroid defect is due to ribosomal defective translation, and what are those mRNAs that are specifically deregulated? And finally, pathophysiology of DBA says P53 is active, and we know that P53 is an anti-tumor response to an animal. So P53 prevents a defective cell to go on to become carcinogenic in nature. So that is why P53 is mutated in 50% of the cancer cases. So if that is to be true in DBA patients, why they end up with cancer later in their life? So the mystery continues. Hopefully, answers will come up, and maybe in future, if I get a chance to share my data with you all again, I'll be happy to answer these questions based on what we do or what we are doing right now. So the take-home message is that the mystery still continues. So I hope I will be able to convince you that with zebrafish, we could do many things, and we could identify a lot of things about the molecular mechanisms of a particular disease that we looked into. I would like to thank the teams that I worked with. I started with Dr. Kemochi's lab in Japan, where I did my postdoc for quite a long time, and I also worked as an independent scientist for some time. And then I moved to France, where I worked with Dr. Glaz's lab in Toulouse, where I was part of the DB registry and worked with patient samples. Of course, I did not show the data that we got from the patient samples because it was outside the purview of this talk. And finally, this is my group here in Nitte. And of course, this photo was taken some time back. The group has increased in number, and we are continuing our research on DBA and ribosomopathies. Thank you very much once again, organizers, for giving me this opportunity. I overshot by 5 minutes. Sorry about it, but I wanted to complete the story as I envisaged or as I planned. So thank you once again for this opportunity.
Thank you, sir. Thank you for such a wonderful talk. I'm sure the students have learned a lot with that. We will go ahead and take a few questions. Amla, can you please continue?
Okay, ma'am. There's few questions by the participants.
Sure.
Ma'am, am I audible?
Yes, you're audible.
Okay, so there are a few questions by the participants. Question one is, what methods were used to knock down RPS19 genes? And sir, can you please clarify, was it knockdown or knockout?
All right. So when we started with morpholino-induced knockdown. That is a transient knockdown that we do. Then we also used a mutant which was developed in Hopkins lab. We procured it, and it is basically a viral insertion mutagenesis. So the mutation, the viral sequence was getting inserted in the ribosomal protein sequence. So we used both a mutant as well as a morphant. So the knockdown was transient, it was not a knockout. Yes, in the mutant, you could say that it was a null mutant, so it was a knockout. Right now, we are working on CRISPR-based mutant, which are much easier compared to the insertional mutagenesis that we used earlier.
So there's one second question saying, it is unclear how missense and nonsense mRNA did not rescue the anemic phenotype, because RBCs are visible in the image, right? So can you please clarify and explain the same?
Okay. I'll go back to the slide. I'll explain it again. All right. Maybe the next slide. Yeah. So here you see on the left-hand side, we have shown the normal fish. So on the upper image, it is the wild-type fish, which was not injected with anything. It is just a wild-type. And if you see that the brown spots, which are a lot more dense and a lot more crowded compared to the ones which are in the middle of the image. So in the middle, you have severe reduction of these brown spots. So that means these are anemic. So in the lower image, missense and nonsense mRNA were not expected to carry on with the rescue because these are patient type mutation. So in patients, RPS19 are mutated in these positions of the gene. So these missense mutations are not, missense or the nonsense mutations are not expected to rescue. So if you see the left-hand image, the brown spots that you're seeing are a lot more compared to on the images that you are seeing on the middle and the last image. Yes, when we talk about rescue, it is impossible to imagine a complete rescue. If it is always sometimes a partial rescue or probably 80% or 90% rescue. 100% rescue is highly difficult to achieve. So here, and we are talking about morpholino injection. So there are many things which are variable. So it is expected that rescue is not going to happen, and that is what we found, that they did not have rescue. So because these are patient type mutations, so if they were to rescue, then that means the model system that we have developed is not recapitulating the phenotype that we expect in the patients.
Thank you, sir.
Thank you, sir. It was a pleasure.
All right. So thank you very much once again. I will stop my screen sharing, and I can see Chetana there already. Hello, Chetana.
Hi, everyone.
Hi. Good to see you. So
Yeah,
sorry I took time. I think I leave it to Dr. Himanshu. Over to you, Dr. Himanshu.
Yes, sir. Thank you, sir. Coming to our next speaker of the day, we have Dr. Chetna with us. I'll just share my screen. Just a second, sir. I have an issue. Just a second. Yeah. We have with us Dr. Chetna Sachidanandam. Madam is an associate professor and coordinator at CSR Institute of Genomics and Integrative Biology, IGIB, Delhi. Madam is a PhD in life sciences from CSIR CCMB, Hyderabad. Madam's research interests. Her lab uses zebrafish to dig deeper into the working of human diseases, and they focus mainly on diseases of the liver caused by genetic mutations, lifestyle, or by drug-induced toxicity, rare monogenic disorders that cause developmental defects resulting in birth defects. Her lab has also discovered new insights and promising therapeutic leads into Charge syndrome, Rubitson's type B syndrome, hereditary hemochromatosis, and liver regeneration. Her current focus is on taking these discoveries towards meaningful clinical applications. Ma'am is going to talk on the lean fat Indian zebrafish, a model for fatty liver disease, revealing new biology. Over to you, ma'am.
Thank you, Dr. Himanshu. So I will share my screen. Please tell me if it's visible when I.
Yes, ma'am, I will. Yes, ma'am, it's visible.
And now it's visible in the slide show mode. The
Yes,
full
ma'am.
slide is visible.
Yes, ma'am.
Okay. Okay. So thank you for this opportunity to give this webinar, e-webinar. This is a lost opportunity to visit your institute, but maybe some other day. So, so when I heard that you're doing a webinar on zebrafish as a emerging model, I thought I will do, and ma'am said that Anirban will be talking first on genetic disorder. So I'm going to talk something different. But what you see on this slide is basically what my lab is interested in, basically modeling human diseases in zebrafish with the goal of understanding more about the diseases and to find drug candidates or genetic ways of rescuing the disease phenotype. So I've just highlighted the diseases that we work on that Madam talked about already. So we work on genetic disorders mainly because that's a very huge strength of zebrafish. But today I'm going to talk about something that is not genetic, more of a, you could say, lifestyle or otherwise. So non-inherited, non-alcoholic fatty liver disease is going to be the topic today. And of course, the system that I'm going to talk about is zebrafish. But I also, because Anirban already gave you a flavor of how embryonic zebrafish can be used to study biology, human biology, I want to talk about adult zebrafish. Today, almost all the talk, the whole talk is on adult animal. And just to give you an idea of the organs and their placements, I have put an image here with showing the a schematic of various organs in the zebrafish. I hope you can see my mouse. So when I point to something particular, we will be able to visualize that.
Yes, ma'am.
So yeah, okay. So so let me go to the next slide. So fish is small, 3 centimeter maximum maybe, and the organs are much smaller than that. So compared to mouse, working with them is much more difficult, but there are a lot of advantages. Anyway, so the title of today's talk is the lean fat Indian fish, and the name comes from the sort of the colloquial name that is used for a non-alcoholic fatty liver disease in people who are not obese. So the lean fat Indian is considered to be people who have fatty liver disease but are not obese. So I'm going to talk about this kind of a disease. But first of all, what is NAFLD or non-alcoholic fatty liver disease? It's basically non-alcoholic, as in not alcohol related. So when you consume alcohol in large quantities, or alcoholics generally have liver damage and fat accumulation in the liver, that is called alcoholic fatty liver disease. When you do not have alcohol as a driving force, then you either have genetics or lifestyle or other associated causes which causes fat accumulation in the liver, and that is called fatty liver syndrome, which is non-alcoholic. And usually from a healthy liver, when the liver becomes fatty or it starts accumulating large quantities of fat, the next step that happens is called non-alcoholic steatosis, steatohepatitis, which is basically more damage, followed by fibrosis, where the liver tissue, liver cells lose their function and more fibroblasts take over the tissue population. Final stage would be cirrhosis, where you have major loss of liver function and can lead to liver failure. So this is progressive liver failure that you're seeing, and the function of liver gets affected more and more. And it's an essential organ. I don't need to tell you that. So the estimates vary from 30 to 50% of NFLD in Indians. This is also very large in other populations. I'm just mentioning the Indian population here. And so, something that is sort of interesting and which is relevant to what I'm going to talk about later is that inflammation always seems to be sort of around when there is NFLD. So patients with NFLD always seem to have high levels of inflammation and high levels of cytokines such as interleukin 6, interleukin 1, TNF alpha, etc. And I've highlighted with a red box here how compared to hepatitis patients or with healthy control liver, you have high levels of TNF, high levels of IL-1, high levels of IL-6. This is also very interesting when you look at general Indian population. So this was a study that performed in United States, and they compared populations of different ethnicity. So I'm showing you only Caucasians and Asian Indians in this slide, but they did Black Americans and South Americans, etc. And they found that the Indian group of healthy, lean, non-smoker young population seems to have a significantly higher levels of IL-6 and significantly higher levels of liver triglyceride levels compared to Caucasians in this picture, but in the paper compared to other populations as well. So it seems that so as I said, inflammation seems to be associated with fatty liver generally. An Indian seems to have a preponderance for having more interleukin 6 and more inflammation. There are some other data that is accumulated, which is also interesting in that diseases where there is systemic inflammation, such as inflammatory bowel syndrome or systemic lupus erythematosus or rheumatoid arthritis, all of these seem to have a lead to or be associated with a large proportion of people who develop non-alcoholic fatty liver disease. So the percentages I'm showing you here are these different studies that were performed, where they showed that 33% of the inflammatory bowel syndrome patients they studied had non-alcoholic fatty liver disease. So there is more and more sort of this very tenuous, very intriguing relationship between inflammation and fatty liver. And what that has to do with the lean Indian, I will talk about it towards the end. So basically, the first question we wanted to address was, does inflammation by itself then trigger fatty liver? Because usually people only see that there is fat in the liver and then they go and look at the patient and they find more inflammation. So you don't really know, did the inflammation cause fatty liver or did fatty liver cause inflammation? And we wanted to sort of parse out this relationship. So we used, of course, zebrafish, and we used a transgenic model of zebrafish to create chronic systemic inflammation. And the model we used was a double transgenic system. So not a knock-in or not a knockout or a knockdown, but rather something that is we are incorporating into the genome. So we had two constructs. One was the myosin light chain, cardiac myosin light chain promoter driving GAL4-VP16 protein. So you must be familiar with the GAL4-VP16. It's a transcription factor activator, which activates transcription by binding to a sequence segment called upstream activator sequence. It's a yeast system which has been adopted for driving gene regulation in higher organisms. So very frequently used in Drosophila. So the cardiac myosin light chain promoter is driving GAL4-VP16 protein. So when the protein is made in the heart only, because the promoter is active only in the heart, that protein then goes to the upstream activator sequence and binds there, and Downstream of this, we have cloned the human IL-6 cDNA. So basically what happens is, in the heart, GAL4 is made. GAL4 leads to the expression of human IL-6 in the heart. So heart is shown in green in this, a different transgenic animal that I'm talking about, just to illustrate where is the heart and the liver. So the heart is here. So IL-6 or interleukin 6 is a cytokine, and it is a secreted protein. So what we wanted to do was make it in the heart, but it will get secreted out of the heart cells and it will go through the bloodstream, and your whole body gets exposed to IL-6. So, and we wanted to study what is happening to it in the to the liver upon exposure to IL-6. But first of all, we want to check whether the system is working or not, right? So we did two kinds of experiments. Here I'm showing you an RNA in situ hybridization of an embryonic zebrafish, the normal controlled zebrafish, probed with a probe for human IL-6. No signal in the heart. Here is the IL-6 overexpressing line that we have created, and you can see nice purple staining in the heart, indicating that the human IL-6 mRNA is being expressed in the heart of these embryos. In the adult animal, we took the heart out after sacrificing the animal, and then took the whole heart and stained it for human IL-6 again. And using the RNA in situ hybridization, we could show that in the male as well as female adult heart, you could see signal of interleukin 6 binding, while the control hearts failed to show any signal. We quantified the mRNA, or we did a real-time PCR, and we saw that in the interleukin 6 overexpressing or IL-6 OE embryos that we are looking at, or the liver we are looking at, we can get signal at much fewer cycle numbers, PCR cycle, compared to the control. So you can see almost a 20 cycle difference between the IL-6 OE fish liver and the controls. We did Western blots to see whether the protein of IL-6 or the small peptide of IL-6 is being produced, and we can again see in IL-6 overexpressing fish heart. Now we are looking at heart here. I'm sorry, this is also heart expression. We are looking at the heart, and the protein is also expressed highly. So we can establish that the human IL-6 is made in the heart. What happens to that IL-6? Does it travel to other parts of the body? So we took the liver and looked for genes that are downstream of IL-6 signaling to see whether there is induction of these genes. So all the red dots here are the IL-6 overexpressing fish. The blue are the control fish. And you can see in the case of each gene, SOX3, MIC-A, hepcidin, 3 genes that we looked at, which are downstream of IL-6 signaling, all of them are induced in the transgenic animal, but not in the control, right? So indicating that although the protein of IL-6 is made in the liver, it is being secreted in the bloodstream, and it is traveling through the body and causing signaling to happen in the liver. So the liver is responding to the high IL-6 levels in the animal. What happens to the liver? So we did histology, first of all. So in the top panel, you are seeing the hematoxylin and eosin stain of controlled male liver and IL-6 male liver. And you can see the difference. I don't need to point it out, that in the male IL-6 liver, you can see a lot of white spaces. It's like bubbles in between. This is indicative of lipid accumulation. This is what fatty liver looks like in human tissue also. And we did a stain for oil redo, which is a red colored stain which binds to oil or lipid. And you can see, compared to the control, the IL-6 overexpressing liver has lots of blobs of red, indicating again lipid accumulation. Same thing, but a different kind of stain. BODP is a stain that is binding to lipids again, but it is fluorescent. So you can see that in the male IL-6 overexpressing liver, you have lots of green signal, which is absent in the control male. Now, the interesting bit also was that the female animal did not seem to have lipid accumulation as we saw in the male animal. Although the transgene is very similar, the expression of IL-6 was similar, I showed you in the previous slide, and the signaling also was very similar. But it does not accumulate lipid. We don't know the answer why. That is something that is pending. It's a story in progress. So we saw lipid accumulation in the liver. We wanted to know what is going on in the liver, what drives the accumulation of lipid. So, okay. So first we also wanted to quantify the lipid accumulation. So we did thin layer chromatography of the livers of control and IL-6 and found that cholesterol accumulation is happening and triglyceride is accumulating. So both of these lipid molecules are there in higher levels in the transgenic animal compared to the control animal. So now to try and figure out what is molecularly happening, we decided to do a total transcriptomics of the IL-6 overexpressing zebrafish liver. So we took the adult liver of male and female. We took 4 of these controls, IL-6 overexpressing, control and IL-6, and we did a total transcriptomics. We found around 1700 genes that are differentially expressed between these samples, which I'm just showing you in a heat map here, just to illustrate to you 2 things that I'm illustrating here. First of all, there is a dramatic difference between control and the IL-6 OE in the male as well as in the female. You can see that there is a very big difference between the patterns that you see if you go just top to bottom like this and top to bottom like this. Second very interesting thing we found was the control liver of male and control liver of female itself is very different. So this might be one of the reasons why we are seeing a very different sort of response to the stimulus that we are giving. When we overexpress IL-6, we are seeing very different patterns of expression in the genes and very different manifestation of the phenotype as well. Anyway, so we looked at the genes that are induced in the male IL-6 liver. And one of the things that we saw was that the JAK-STAT signaling, which is the signaling process pathway downstream of IL-6, is induced, which was very nice sort of confirmation that the signaling of IL-6 that is released from the heart is happening in the liver and the liver genes are responding to it. Then we looked at what are the interesting patterns of expression that we are seeing in the total transcriptomic signature that we are seeing here. So one of the things that jumped out at us was that a large number of genes of the glycolysis gluconeogenesis pathway were downregulated. So I want you to look on the right side where there is a heat map. Just for reference, this is slide number 15. So the control is showing you red colored blocks, and that is indicating high levels of gene expression compared to the transgene, which is showing you blue blocks, which are showing lower levels of gene expression. So it's a comparative plot that I have plotted here, and you are seeing that there is a downregulation of a number of genes that lie in the gluconeogenesis pathway, which I have highlighted on the left side in the pathway with all the blue enzymes are basically the RNA that is coding for these enzymes are downregulated in the transgenic animal. We wanted to see if this is true for protein levels as well. When we did a proteomics experiment with the same livers of male and female IL-6 and control, we found a bunch of proteins that were up or down regulated, but something that was very interesting was this aldolase B gene that is here, which converts fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate or back. It's a bidirectional gene enzyme. And we saw that there was a very nice downregulation of aldolase B protein as well. So this was a gene that was downregulated in the RNA level as well as protein levels, right? So what happens if when you have deregulation or perturbation of genes that code for enzymes of glycolysis gluconeogenesis pathway? You would expect that there would be metabolic disturbance, right? So we did a metabolomics of the livers of control and IL-6, and we got a large number of metabolites, of course, including some lipids. But it was very interesting that we got a large number of metabolites of the carbon metabolism pathway, and on the left hand you see the glycolysis gluconeogenesis pathway. On the right hand side there are other hexose phosphate conversions that are happening. So I have shown you that from glucose 6-phosphate there are many other sugar phosphates that get converted for various purposes in the body, and there is disturbance in these two. So the orange colored metabolites are increasing in level or accumulating, and the green colored ones are decreasing. And in this picture, the blue enzymes are all going down in the transcriptomics, and the red enzyme is going up. Okay. So we saw disturbance in the metabolites as well. So basically, what we think is happening is here. So it was very interesting that there is this pathway that happens, a step that happens where the glyceraldehyde 3-phosphate and dihydroxyacetylene phosphate are basically same molecular weight, same molecules with slightly different structures, and this TPI1B isomerase flips them. So we see that there is an increase in this protein level, TPI1B in the from the proteomic data, and there is an increase in the GPD2, which is a protein enzyme that converts dihydroxyacetone phosphate to glycerol 3-phosphate, and glycerol 3-phosphate is one of the precursors for creating triacyl glycerol. So this is where we think the triacyl glycerol is coming from. Basically, the deregulation that you're seeing in the glycolysis gluconeogenesis pathway is leading to an accumulation of triacyl glycerol through this shunt that is happening here. And there was more support to the data that so our data suggested that the inflammation is causing accumulation or decrease in many enzymes of the glycolysis gluconeogenesis pathway, and that decrease is leading to accumulation of various metabolites, which are now being shunted into a triacyl glycerol synthesis pathway. So something that when we found this interesting data, we went looking for, is there anything known about these associations? And we found an interesting paper that was not too long ago, last year, that showed that patients with the genetic disease called fructose intolerance, which is caused by a mutation in aldolase B. So they have aldolase B deficiency or inactivity in these patients, and because of that they are intolerant to fructose. We found that the patients with aldolase B deficiency have a higher intrahepatic triacylglycerol content, which suggests that we are seeing an aldolase B downregulation in our fish, and these are human beings who have a natural situation where there is aldolase B downregulation, because of which they are having a higher level of hepatic triacylglycerol content. So similar to fatty liver content, right? Then we went about to see whether anything else is known in fatty liver disease and aldolase B regulation. So we found a data set where they had looked at hepatic transcriptome signatures in patients with different levels of fatty liver. So normal people, obese people, non-alcoholic fatty liver people, and non-alcoholic steatohepatitis. So different severity levels. And their data set, we took the data set, we reanalyzed the data set and looked for aldolase B expression in this patient group. And we found a nice distribution. If you see here, this is the analysis we did. So the normal and obese people have similar kind of levels of aldolase B. However, non-alcoholic fatty liver people and NASH, non-alcoholic steatohepatitis patients, both of them had significant downregulation of aldolase B gene expression in there. And this was a data that was not looking at this gene at all. This was an independent data generated by different people for different purposes. And we could see that there is actually an aldolase B downregulation. So this all gave us sort of confidence that we are sort of what we are seeing might be relevant to human biology. So now I will kind of lead you to our model, what we think is happening. What we think is happening is that the hexose phosphates are getting converted to dihydroxyacetone as well as 1,2-bisphosphoglycerate, different pathways in the glycolysis gluconeogenesis. Then the enzymes that kind of do this conversion are downregulated. Because of that, one side of it, we don't know why the hexose phosphates are also not causing a different kind of shunt, but the DHAP or dihydroxyacetone phosphate and glyceraldehyde 3-phosphate is accumulating, which is getting converted into glycerol 3-phosphate by this enzyme GPD2. This glycerol 3-phosphate becomes the trigger for the raw material, the building blocks for making triacylglycerol, which accumulates in the liver in large quantities. So, and we think that this is induced or this is driven by IL-6 because our control animal, which has everything else, the inducer of the Gal4 BP16 is there, but there is no interleukin-6 in our control fish, and those fish do not accumulate triacylglycerol, while the fish that are exposed to IL-6 continuously over long periods of time do accumulate TGs and their liver. And this IL-6, how it induces these genes or downregulates these genes, we have some clues. We have found binding elements on the aldolase B promoter, for example, for IL-6 signaling components as well as PPAR gamma T signaling components, which we think might be important for this regulation of aldolase B by IL-6, but we still have not done the experiments to prove that yet. So that's the next level. But this is what we think is happening. And how is this interesting for lean fat Indian concept? I already showed you that the lean Indians seem to have a higher level of interleukin-6 generally. So there is a high baseline systemic inflammation in the Asian or the Indian East Asian population, the Indian subcontinent population, compared to other kinds of ethnicities. And this higher level of systemic inflammation might be the cause for triggering fatty liver, in spite of the fact that the patients do not have obesity. So usually all the animal models that people study to understand fatty liver or diabetes, both of them concentrate very sort of unequally on obesity-driven disease, obesity-driven metabolic syndrome. So the mouse models, all the human studies focus on why obesity or adiposity causes triacylglycerol accumulation. We have a model now which allows us to look at something that is independent of the diet or independent of the obesity. These fishes that we have do not have extra fat content in their body, are not obese, do not feed more than the other fish. So there is no, for example, leptin mutation causes fish to or animals to overfeed and become very fat. We do not have any such complications, except that there is systemic inflammation. This systemic inflammation seems to lead to a deregulation in the metabolism that is causing triacylglycerol accumulation. So I'll summarize my talk by sort of telling you what all I covered here. Basically, what we did was we created a chronic systemic inflammation model in zebrafish, which allows us to study in detail what is going on inside the organs. This inflammation seems to cause fatty liver in the male animal, not in the female. The fatty liver is associated with deregulation of glycolysis and gluconeogenesis. There is a metabolic imbalance because of this gene regulation defect. And this metabolic imbalance leads to triacylglycerol synthesis in the liver and accumulation. We think that this suggests a possible mechanism for lean fatty liver disease. And one of the things that we would like to do in future, this is the power of zebrafish, is to use small molecule screens to identify molecules that can revert this behavior, this phenotype, so that we can identify chemical molecules and their targets that can actually change the behavior of the fish or the lipid synthesis in the liver. That way, try to uncover more new biological insights into the disease model that we are looking at. Finally, I will acknowledge the team that worked on this story. Basically, Manoj, who's my PhD student, this is his PhD project, sort of from beginning to end. And we had a lot of help from within the institute, where we did the transcriptomics and proteomics, and a lot of colleagues and their students helped us. We also had help from CSIR NCL, where we performed the metabolomic experiments, and from AIMS in New Delhi, where we got a lot of help in understanding the phenotypes and interpreting our phenotypes. So I'll conclude here and take questions.
Thank you, ma'am. Amla, do we have any questions here?
No, ma'am, we don't have any questions. We can continue.
Okay. Thank you, ma'am. Thank you,
Thank
ma'am,
you.
for such an informative talk. It was really down to it. I personally will really get in touch with you for my further research.
Okay,
It
sure.
was really helpful for me.
Thank you.
Thank you, ma'am. Coming to our next speaker, we have with us Dr. Patrick Sips. Sir is a faculty of medicine and health sciences from Department of Biomolecular Medicines, Ghent University Hospital, Belgium. Sir obtained his PhD in science from the Ghent University. His postdoctoral research project was on mouse cardiovascular physiology at Massachusetts General Hospital and Harvard Medical School in Boston, where he was later on trained to use a zebrafish as a model for cardiovascular diseases at the lab of Professor McRae at the Brigham and Women's Hospital, Boston. Sir is working on connective tissue research group at the Department of Biomolecular Medicine, Ghent University. Sir's research interests are on preclinical animal model research projects, primarily focusing on connective tissue disorders with cardiovascular complications. Sir is going to talk on heritable connective tissue disease modeling in zebrafish from patient to aquarium and back. Over to you, sir.
Okay, thank you very much for the introduction. And also I'd like to thank the previous speaker, Chetana. I'm sorry that I joined in a little bit late, but it was very nice to see your results and very nice to actually see you again after we have met. Maybe you remember approximately 10 years ago when I was also at the MGH in Boston when you were there.
Yeah,
good to see you again and congratulations on your work.
Thank you.
So let me just see if I can share my screen and then I'll show you a bit of what we are working on. Okay. All right. So as was mentioned in the introduction, of course, I'll tell you a little bit about our work on heritable connective tissue disease that we are doing in our Center for Medical Genetics in Ghent. I'll start with a very quick background just to have an idea of the, basically the embedding of our center. So we are situated at the Ghent University Hospital, which is the second largest academic hospital in Belgium. And of course, our center has an important diagnostic role where we do routine panel sequencing, whole exome sequencing of patient samples, pre-implantation genetic diagnosis, non-invasive prenatal testing, as well as genetic counseling, of course. But as the title probably suggested already, we have a very strong interest in heritable connective tissue disease, which is something that actually grew since the connective tissue disease lab was founded in 1986 by Professor De Pape, and which has grown significantly over the past years with the help of several other clinical geneticists who joined the group and have expanded the research into connective tissue disease. So very briefly, to give you a very, let's say, wide overview of what is connective tissue disease or connective tissue per se. So you can consider connective tissue as sort of the glue that holds us together. It's basically consists of a complex extracellular matrix containing collagens, microfibrils, glycosaminogens, and a lot of other molecules as well. Of course, the main function of connective tissue is to provide structural support to tissues, but it's also playing a role in intercellular communication. And it is important in most organ systems, actually, I think almost all organ systems in the human body. This also reflects a little bit in the effects of diseases of connective tissue. So as I mentioned, we focus in our genetic center pretty strongly on the heritable connective tissue diseases, which are usually very rare genetic disorders with prevalences ranging from 1 in 10,000 to 1 in 1 million, or even lower sometimes. And we basically have 5 areas of focus in our center. On one hand, one disease we're focusing on is osteogenesis imperfecta, which is also called brittle bone disease, and has, as you can see, very severe bone disorders. We also study Marfan syndrome, which apart from skeletal phenotype also shows very strong cardiovascular complications, which can be life-threatening. We also study pseudoxanthoma elasticum, which is characterized by ectopic calcification in the skin, but also in blood vessels. Then there is cutis laxa, which is manifested as basically laxity of the skin, as the name suggests. But it also has a wider impact on other symptoms such as craniofacial disorders, intellectual disability, and also cardiovascular complications. And a final syndrome we are looking at is Ehlers-Danlos syndrome, which is also manifesting itself as skin laxity, but there's also a hypermobility component, and patients also often are prone to easy bruising and can also present with cardiovascular complications. So there are several important clinical challenges for managing these heritable connective tissue diseases. On one hand, for still a quite significant number of patients, the genetic cause is still unknown. We also see very large phenotypic variability within patient groups of the same disease. And of course, a big problem is that for basically all of the diseases I've shown thus far, there is no cure available, so we can only provide supportive treatments. So what we would aim to do is to try and identify and validate a new potential causal genetic variants to be able to help with the diagnosis of, or at least genetic diagnosis of patients of connective tissue disease. We also want to explore the role of genetic modifiers, which play a role in regulating this large variability that we observe. And we also want to elucidate the pathogenic mechanisms behind the disease and eventually test new therapeutic compounds. So in order to be able to perform all these goals, there's clearly a strong need for flexible functional models. And this is something, of course, that has also been recognized in the connective tissue disease research group, where in 2010, actually, they started with the first zebrafish rack in the lab. And this has steadily grown, where up to the point where last year we have officially founded the Zebrafish facility Ghent, which is now a core facility of the university. So our facility is a medium-sized facility of 17 zebrafish racks on semi-closed recirculating systems from Techniplast, with a capacity of approximately 20,000 adult zebrafish. So the users are not only the people from the connective tissue research lab, but also people from cancer genetics and neurogenetics are using zebrafish as a model in our institute. So I think I don't really have to show the slide anymore, considering the previous speakers, but I would still like to highlight a few of the benefits of working with zebrafish, and then particularly zebrafish embryos. As you can see, these embryos develop, of course, they develop externally, and they are translucent during the first days of development. So they can be easily observed using very simple microscopy techniques. They can be grown in 96 cell plates for the first days of development, so they can be used for drug screens. And what is also important is that it's quite straightforward to manipulate their genome or to produce transgenic lines. So we can, I mean, in the past, most use has been made of the random ENU mutagenesis lines that have been characterized over 20 years ago, approximately. Then people started working more with morpholinos for targeted gene knockdown. But these days, I think most of the field has transitioned to specific gene knockout using CRISPR technology. And I think Talon has already seeing a bit more limited use at this point. And as I mentioned, we can easily overexpress transgenes to make new fluorescent or luminescent reporters. As I mentioned, we can non-invasively monitor many phenotypes in zebrafish during development. For example, we have several tools at our disposal to look at cardiovascular function. On the top, you can see some examples of bright field microscopy images or videos of zebrafish embryos that are 2 days old. So on the left, you can see the beating heart of the fish, where you can basically also extract a number of parameters, such as the cardiac function of in this fish. And on the right, you can see the blood flow inside the tail of a zebrafish, which you can track. These videos are slowed down, of course, a little bit because the heart rate is higher than what you see here. We can also explore the structural properties of the vascular system using fluorescent reporter lines, such as in this case, the KDRL GFP, which has an endothelial-specific overexpression of GFP. And in the bottom is an example of a transgenic line showing fluorescence in erythrocytes. So you can easily track individual erythrocytes as they circulate throughout the body. Similarly, there's tools available for looking at bone malformations or bone phenotypes. One line we have in our lab is the Ostrix guidi line, which specifically labels osteoblasts in the zebrafish. On the other hand, we can also use staining techniques to look at different components. So we can use Alcium blue to look at cartilage, or we could use alizarin red to look at mineralized or calcified tissues in the zebrafish embryos and larvae and beyond. We also have been working on optimizing a number of phenotyping tools for adult zebrafish as well. So we have been working on cardiac ultrasound to optimize this technique for zebrafish. As you can imagine, this is not straightforward considering the size of the zebrafish, as they are only a few centimeters large, and the heart itself is approximately only 1 to 1.5 millimeter diameter. But we can actually observe the heart using the correct high-frequency probes. And here's an example of a beating zebrafish heart where I have outlined the ventricle of the heart. Recently, we also have begun optimizing a new technique to perform vascular corrosion casting, where we basically inject a liquid polymer solution, a polymerization solution, which will then solidify inside the vasculature of the zebrafish. And then we can look at the actual structure of the vasculature if we remove all soft tissue from the fish. Of course, this is a post-mortem analysis. Obviously, this is still in vivo. Also for bone structure, we have several tools that we can use. There is, as I mentioned, alizarin red, which can also be applied to the adult zebrafish, where you can nicely see all the calcified bone tissue. But we can also use more advanced techniques, such as the synchrotron, which we can use in collaboration with a lab in Switzerland, where you can closely look at the phenotypes of individual vertebrae, for instance. Or we can use micro-CT to have an idea of the general skeletal structure in the zebrafish. So during the course of the years, we have been working on obtaining zebrafish models for these disorders. And we have actually, at this point, successfully generated fish models for pretty much all of the models or diseases we were interested in. And to a pretty large extent, they show relevant phenotypes which reflect what is actually been seen in patients. So for today, I would just like to focus on osteogenesis imperfecta and Marfan syndrome as two examples. Otherwise, I think it will take up way too much of your time. So to reiterate a little bit the background on osteogenesis imperfecta. So as I mentioned, this is actually called also the brittle bone disease. It's characterized by pretty significant clinical inter and intrafamilial variability. As you can see, it can range to very mild phenotypes to very severe ones where people are severely disabled. So the collagen, of course, is a major constituent of bone, and it has been shown that pathogenic variants in the genes coding for type 1 collagen are linked to various types of osteogenesis imperfecta. As I'm showing here already, this is the say, the basic structure of a type 1 collagen, which is a triple helix consisting of 2 alpha-1 subunits and 1 alpha-2 subunit. We have investigated this in zebrafish and actually found that the COL1A1 gene is duplicated in zebrafish. So there's an A and a B isoform. So in zebrafish, there's not only alpha-1 and alpha-2, but there's also an alpha-3 protein. But apparently, these three, they combine also to form a similar triple helix as can be seen in humans and other mammalian organisms. So then we have started generating, or we have obtained certain existing mutants and have generated some new mutants in-house. And we found that in general, the null alleles caused more of a mild form of OI, whereas the missense mutations caused more of a moderate or severe OI. So to be able to capture the entire phenotype of the zebrafish, we make very strong use of the micro CT scanning facility, which we can use in collaboration, which helps us to really get a nice overview of the complete bone structure in the fish, as I mentioned before. So this is an overview of the different mutations we have studied and that we have actually in-house in our department. And I'd just like to briefly go over a few aspects of these, the phenotypes that we see. So if you look at the fish which have a null mutation in one of the collagen Nicol-1 genes, this seems to be most like a mild OI type 1 phenotype. So we see only very small abnormalities, such as vertebral compression and some abnormalities in the other bone structures. But what is interesting is that we also saw a pretty significant variability even within the same genotype, which is again something that kind of recapitulates what is being seen in human patients of OI as well. Another mutant that was interesting was the COL1A2 mutant, which seems to be more reminiscent of an OI / Ehlers-Danlos syndrome phenotype. As you might recall from the introduction, Ehlers-Danlos syndrome is more of a soft tissue syndrome where patients show this skin laxity and hypermobility of the joints. And we actually also identified this in our COL1A2 mutant fish, where we did see soft tissue phenotype. As you can see, the skin is clearly shows malformations where there is less collagen staining in the skin of the adult fish. And we also saw these certain skeletal malformations as well in these COL1A2 mutant fish. And then, as I mentioned before, these are all the missense mutants, where glycines have been mutated to different amino acids. And these seem to correlate to the more severe, moderate to severe OI types. So these mutations of glycine most likely disrupt the potential of the collagen subunits to form these strong trimers, which leads to perhaps more of a dominant negative effect, which is more detrimental to the phenotype of the bone. So we can see a number of phenotypes here, such as a shorter axial skeleton, vertebral compression, scoliosis, distorted vertebrae, overmineralization, and fractures as well in the fish, which are all symptoms that are also expressed by patients of OI. So just to briefly summarize this part, this study has actually been published in PNAS recently, and was the first phenomic study of a relatively large cohort of different zebrafish mutants which were related to a human brittle bone disorder. We found that the spectrum, the phenotypic spectrum of these zebrafish mutants quite strongly mimics the spectrum of human OI, including also this variability that was seen not only between different genetic mutations, but also within the same genotype. So this would be probably a promising tool to try to understand the molecular basis, which drives the clinical phenotype, and also this variability that is seen in OI. And on the other hand, we also showed in this COL1A2 mutant, this first zebrafish model for EDS, for the soft connective tissue EDS disorder. In the second part, I'd like to switch gears a bit and talk to you about Marfan syndrome, which is actually the subject I've been working on more specifically myself. So Marfan syndrome is a connective tissue disorder, obviously, which has a prevalence of about 1 in 10,000, and is caused by dominant mutations in the fibrillin-1 gene. What is quite clear, strongly seen in these patients is not only the skeletal abnormalities, so obviously often you see overgrowth of large bones in these patients, but there's also a very strong cardiovascular component of the disease. And actually, this is the major cause of morbidity and mortality, as these patients present with aortic aneurysms, which can progress to dissections and ruptures, which of course can be life-threatening. And on the other hand, there's also evidence for primary cardiomyopathy in Marfan syndrome patients, and perhaps even for increased arrhythmia. So fibrillin-1 is a very large gene and contains, well, so far over 3,000 pathogenic FBN1 variants have already been characterized. But a large number of variants that are identified in the clinic are still characterized as variants of unknown significance, meaning we cannot predict whether this variant actually causes disease or not. And as with the other connective tissue disorders, currently only supportive treatment is available. So patients will be usually put on antihypertensives to prevent severe complications, but no real cure is available. So again, this reiterates the use or the need for a good functional animal model to be able to address these issues. So to begin to make a zebrafish model for Marfan syndrome, it's of course important first to find the right gene to target, which is can be a little bit tricky sometimes. In humans, there are 3 fibrillin genes. As I mentioned, fibrillin-1 causes Marfan syndrome, while the other 2 genes, so fibrillin-2 causes contractural arachnodactyly, and fibrillin-3 is more involved in very rare disorders, such as polycystic ovary syndrome. But fortunately, in the fish, there's also 3 isoforms. However, the orthology between those 3 is not entirely clear yet. You can see that even within the human genes, there's a very strong similarity between the 3 fibrillins. So they are also structurally very similar. Which is also sort of hinting already to the fact that most likely there might be some redundancy or they might compensate for each other. But there is one specific region between around amino acids 390 to 450, where you can see more of contribution of proline in the fibrillin-1 protein, more glycine in fibrillin-2, and a mixture of both in fibrillin-3. So if you compare these properties with the predicted amino acid compositions for the zebrafish fibrillins, it seems like fibrillin FBN1 in zebrafish might be the ortholog for FBN1 in the human. Although for the other 2, it's not particularly clear, but we would assume that FBN2B might be the ortholog for FBN2 based on these properties of the protein. We also did, of course, some more clustering of the entire amino acid sequence. And as you can see, while there is, of course, some divergence, it seems that fibrillin-1 from the fish clusters most closely to fibrillin-1 in the humans, whereas the other 2 fibrillins are a bit more separate. Fibrillin-2B particularly seems to cluster a bit more separately, let's say, which is something I will get back to a bit later in the presentation. So, of course, we decided then to target zebrafish FBN1. So we used a CRISPR-Cas9 approach to produce mutants in this gene. So we have targeted 5 different regions, or actually sites in the gene to make different lines for fibrillin, to make different fibrillin-1 deficient lines. Unfortunately and unexpectedly, we found no strong cardiovascular symptom in these fibrillin-1 mutant zebrafish. So what I'm showing here is the ADRL GFP transgenic zebrafish, which show GFP in endothelial cells, allowing us to look at the cardiovascular or the vascular, the vascular structure in the embryos. And as you can see, there is no appreciable difference between the wild types and the mutants at any location in the cardiovasculature, or also at different ages during development. I'm not showing this here, but we have made several different CRISPR lines, and none of them showed a strong cardiovascular phenotype in the early stages, which is surprising considering that in humans, so fibrillin-1, as I mentioned, causes Marfan syndrome, but it causes this as a dominant mutation, which means there's always still a second allele of wild-type fibrillin-1 present. If you lose fibrillin-1 completely, this is not viable in humans. So complete loss of fibrillin-1 is lethal in very early stages, which is why it was very surprising for us to find no phenotype in these mutant fish. So then we looked also at the expression of the different fibrillin genes in the fibrillin-1 mutant zebrafish. We did find in at least a few of these lines nonsense-mediated decay of the mRNA transcript of fibrillin-1, but we did not find upregulation of the other fibrillin isoforms in the FVN1 mutants. So there is no real evidence that there is any genetic compensation by these other isoforms. So this could not really explain why we don't see a phenotype. We then also looked at the age-dependent expression levels of these different isoforms, and we found that, especially during the very early development, fibrillin 2B seems to be the predominant isoform, while fibrillin 1 and 2A catch up during development, and especially during adult phases. This seems to be the more predominant isoform. So considering the contribution of fibrillin 1 to the adult composition of fibrillins, let's say, we wanted to look at the phenotype of the fibrillin 1 mutants when they were in adult stages. As I mentioned before, we have developed capacities to look at cardiac function in these adult zebrafish using an ultrasound setup. So the setup is pretty much like this, where the fish will be submerged and the probe is then put on top of the zebrafish, and then we can look at the cardiac function or the cardiac images from different angles to make the specific measurements. Something I should probably mention to make sure everyone is on the same page. Of course, the zebrafish heart is a little bit different from the human heart or mammalian hearts. Obviously, zebrafish only have one ventricle and one atrium, since they do not have lungs, but they rely on gills for oxygen and carbon dioxide exchange. And the other thing that is a bit more peculiar about the zebrafish is that they have this extra structure where the ventricle basically changes into the aorta, where the aorta sprouts from the ventricle, which is called the bulbous arteriosus, which is a very distensile structure, which allows for dampening of the pulse pressure coming from the heart. And most likely the function is to decrease the pulse pressure that is experienced by the gills, which are the first organs to actually receive blood coming from the heart, because then at this point oxygen is exchanged. But anyway, so important to remember is that there is only one ventricle and one atrium in the zebrafish. So if we look at this, we can see the ventricle of the fish, as I mentioned before, and we can trace this, so we can calculate systolic and diastolic volumes and then make calculations of cardiac function and cardiac structure. So we did this for our mutants, of course, and it might be a little bit difficult to see from this video because the phenotype is quite mild. But there is apparently a mild dilatation of the ventricle in the FBN1 mutant lions. So we quantify this over a number of fish and with also a number of different fibrillin-1 mutant lions. And we found this reproducible but mild dilatation of the ventricle, both in systolic and diastolic stages. So this shows that there is at least a mild dilated cardiomyopathy in our fibrillin-1 mutant fish, which is interesting because this is something that also occurs in Marfan syndrome patients. And also in a Marfan syndrome mouse model that we have studied in-house, there's also evidence for at least some dilation of the heart. So this is something that does recapitulate a phenotype seen in humans, although we still cannot explain why we don't see a strong arterial phenotype yet. So this led us to wonder how the orthology of the zebrafish versus human fibrillin genes is actually can be considered. So we like to think of it more of as a functional orthology instead of strictly sequence orthology. And if you look at it this way, you can see, as I mentioned before, that Marfan syndrome is caused by fibrillin-1 mutants, and congenital contractural arachnoidectomy is caused by fibrillin-2 mutants in humans. But that's of course, excuse me, I meant to say this is in the case of variants which cause pathogenic variants in a single allele, so the dominant effect. And if you lose fibrillin-1 function completely, this is lethal, as I mentioned before. This is the same for fibrillin-2. Fibrillin-3 loss is viable and leads to this polycystic ovary syndrome or Bardet-Biedl syndrome. Then if we look at the zebrafish, what happens if we completely knock out the different fibrillin isoforms? So in the fibrillin-1 homozygous mutants, we only find this mild adult cardiomyopathy. Which led us to also look at the other two isoforms. So then we also looked at the FBN2A isoform, and we already made a double fibrillin-1, fibrillin-2A homozygous deficient zebrafish. And we found basically the same phenotype as in the fibrillin-1 mutants, where we saw no larval phenotype, neither in the single or the double knockouts. They both survived to adulthood. And again, we see this, this mild diastolic dysfunction, which we also saw in the, excuse me, well, this is, we see a different phenotype, excuse me. We do see a diastolic dysfunction in the FBN2A mutants, but we don't see a strong arterial phenotype yet, as we would expect from a proper Marfan syndrome model. So that actually led us to look at the FBN2B mutants, which is the third isoform in zebrafish. These have actually been studied before because there have been some mutant lines that have been discovered using the random ENU mutagenesis screens that were done in the late 90s. And at that stage, two lines were identified called Puff Daddy and Scotch Tape, which showed quite severe phenotype, where not only defects were shown in the hearts, but also notochord and the fin fold showed some defects. And in the heart specifically, there was evidence for endocardial, well, impaired endocardial cell proliferation and cardiac development, and also abnormalities in the cardinal and caudal veins of these zebrafish. Which is shown here using these fluorescent lines that I've shown before. You can see in one of these mutants, you can see the dilatation of the caudal vein, which was quite striking. So this led us to think that perhaps FBN2B might actually be the functional ortholog for fibrillin-1, because at this point we do see a much stronger cardiovascular phenotype also in the early stages. But a little bit of a caveat, I would say, using these ENU mutants is that it has been shown, at least for the Scotch tape mutants, that not only fibrillin-2B is affected, but also a different gene has been mutated during the random mutagenesis process. And this gene was apparently linked to fibrillin-2B. And it turns out that this also contributed to the phenotype observed in the mutant lions. So it might be a little bit difficult to use these ENU mutants to properly study the function of fibrillin 2B. This is why we decided to generate our own mutants. So again, we use the CRISPR approach to make new knockouts, basically, of fibrillin 2B. This is where the mutation of the Puff Daddy ENU mutant line was found to be, which causes a frame shift and leading to, of course, a truncation of the entire protein. And we made a CRISPR target in the same exon and confirmed that we actually find, so we found then a mutation and we then proceeded with a line containing a 4 base pair frame shift mutation in exon 5, also causing, of course, a very similar situation as in the ENU line. What's interesting is that we can recapitulate most of the phenotypes shown in the Puff Daddy ENU line. So we also see pericardial edema, we also see the fin fold atrophy, but we don't see the phenotype of the notochord. So in the ENU line, the notochord was very wavy and not straight, not nicely developed. But that is not the case in our line. So that shows indeed that we are most likely having a more targeted effect of loss of fibrillin 2 function. But what's interesting is, again, we see a strong cardiovascular phenotype. We do see a dilatation here of the posterior caudal vein, and we see lethality. Of course, these zebrafish do not survive. They die at approximately 6 to 8 days post-fertilization. So we propose that this might be an embryonic phenologue, which means an orthologous phenotype, perhaps for Marfan syndrome. Interestingly, also in these FBN2B mutants, we found quite strong variability in the phenotype. We saw that 50% of the fish do show this cardiac edema and very severe phenotype. But about half of the homozygous mutants do not develop cardiac edema. They don't show an endocardial phenotype. They do not die at the early stages. They actually survive to adulthood. And, but they still do show the tail phenotype. As you can see here, the fin fold is still atrophied in these zebrafish. So these are some images to show you what is actually happening inside the heart. So what we think is happening in the fish that do show this pericardial edema, that is something that has been shown by other groups as well, is that there is a detachment of the endocardium. So what you're looking at is here is the ventricle and the atrium of the zebrafish hearts. And again, we're using the KDRL GFP line showing GFP in endothelial cells. So in this case, the endocardium lights up. And you can see this is nicely covering the inside of the atrium in the control fish, in the wild-type controls. If you look at the mutants, you can see this endocardial line or endocardial covering or lining of the atrium has completely detached and actually seems to be obstructing blood flow, causing vascular embolism. And we assume that this blocked blood flow leads to the pericardial edema and, of course, ultimately mortality in these zebrafish. But then we also decided to look at the phenotype in the fish that do not show this edema. And what was very interesting to find is that they develop rather normally. So the cardiovascular structure develops normally. But if you look closely here at the, so again, this is the heart. If you look at the bulbous arteriosus, if you remember, this is a structure that is just posterior to the ventricle, which can be correlated to the aortic root in humans. This seems to be dilated. We have actually quantified this, and we could show that the ventral aorta is slightly dilated, but specifically the bulbous arteriosus, or the, let's say, the correlate of the aortic root in humans, is dilated in the zebrafish, which is very similar to the hallmark phenotype that is observed in Marfan syndrome patients. So this strengthens us in believing that actually the FBN2B mutants are a very good correlate or model for Marfan syndrome that we can study further. So one of the goals we had was to use these fish to perhaps go to drug screenings at some point. To be able to do drug screens, it's interesting to have a readout that you can use in high throughput. So we then based ourselves on an existing luciferase reporter line. This was generated a while ago in the lab of Dr. Callum McRae in Boston. And it relies on the fact that the natriuretic peptide B promoter is turned on in cases of cardiac stress. So then a transgenic fish was made, which expresses firefly luciferase upon induction of this promoter. So now you have a model where you have increased luciferase expression when the cardiomyocytes are stressed. And this was validated by chronic injection of isoproterenol or knockdown of troponin type 2, which all caused an increase in luciferase readouts in the embryos that were transgenic for this reporter line. So then we crossed these reporters to our FBN2B deficient fish, and we found that there is a strong induction of this luciferase reporter, and we could see at 5 days post-fertilization a very significant increase in the luminescence readout. And interestingly, there was no real significant difference between the readouts in the fish that showed no edema versus the ones that did show this strong edema phenotype and endocardial detachment. So even the zebrafish that have the, let's say, more milder phenotype of only the aortic dilatation still show upregulation of NPPB luciferase, which is of course very interesting and makes this very useful for screening potential drugs that can modulate the phenotype. So we have tested this also at different ages, and we found that there is already, even at 3 days post-fertilization, there is an increase in the luciferase signal, but it becomes most strongly reproducible starting at 5 days post-fertilization. So as I mentioned, our next objective would be to use these zebrafish in a high throughput chemical screen. So the setup would be relatively straightforward, where we plate the mutant embryos in 96 cell plates, treat them with compounds from drug libraries, expose them to these drugs for a certain amount of time, and then read out the luciferase assay or the luminescence measurements. And then this should be able to provide us with a value, which we can then compare to the controls receiving no treatments. And then if we find fish that have a reduced luciferase signal, this would suggest that their phenotype has improved, and this might be an interesting drug candidate to follow up on. So I'm going to stop here because this is pretty much at the stage we are now in our research on this model. So just to briefly reiterate what we have found, we found that zebrafish have 3 fibrillin genes similar to humans and other mammals, but the functional orthology has not been completely established. We found that FBN1 mutants do develop normally and demonstrate only a mild dilated cardiomyopathy in adulthood. But the FBN2B mutant fish do have a very strong cardiovascular defect and actually do show phenotypes that are relevant to Marfan syndrome. Interestingly, we also found this phenotypic variability in the mutants, which is also very relevant to Marfan syndrome, as patients in Marfan syndrome can really range from very mild symptomatic to very severe life-threatening symptoms. And as I just showed, I believe that this model is useful for in vivo high throughput screening, which is something we aim to start with soon. And the general conclusions of our, let's say, our work in our department is that we can use zebrafish as a flexible model to model these rare genetic diseases. We can recapture human symptoms in our mutant zebrafish models. And as I showed for Marfan syndrome, sometimes we can actually use phenologs or autologous phenotypes as a readout for the model. And of course, it is important to always be aware of the proper genetic orthology or redundancy in case of multiple isoforms or even duplication of certain genes, which happens sometimes in the zebrafish. And as I promised from actually the title of my slides, of course, the goal is always to go back to the patient. So we envision our study as pretty much a cycle where we start from the patient, we identify perhaps interesting pathogenic variants, which can then be tested in zebrafish to identify new causal genes. We can also identify genetic modifiers in our zebrafish models. And as I just mentioned, we could use them to produce, do high throughput drug screening for drug discovery. And of course, the goal would be to bring all this back into the clinic where we can then improve diagnosis of patients of these diseases, improve the counseling, and hopefully also improve their treatment in the future. So with this, I would like to acknowledge all the help from the people from our department. And thank you for your attention, and I'll be happy to answer questions if there might be any. Thank you.
Thank you, sir. Thank you for your enlightening talk. Amla, can we go ahead with a couple of questions?
Okay, ma'am. Sir, there are a few questions asking. One of which is how did you fix the zebrafish while taking ultrasound image?
So can you just repeat what was the question exactly for ultrasounds?
Hello.
Can you repeat the question just very quickly?
How did you fix the zebrafish while taking ultrasound image?
How to fix them is the question, right?
Yes.
Yeah. So the picture I showed you is that was actually what we did in the beginning. So it's a very simple setup where you use a small sponge, basically, where you make an incision and then the zebrafish is fixed in there. That was our initial setup, but there are issues with this because the sponge itself produces a certain amount of pressure on the zebrafish. And we know that the blood pressure inside the zebrafish is very low. So if you exert too much external pressure on them, you actually will block blood flow, which is something we observed if we did Doppler measurements. So then we actually made an in-house small device, which we 3D printed, which keeps the zebrafish in place. It's a little bit of a spring-based system, but we can also adjust the pressure. So the zebrafish are only held in place very lightly without too much external pressure. So we can have accurate results that are not skewed by this, let's say, too much pressure on the outside of the fish.
So there's another question asking what is the exact heartbeat rate of 72 HPF in zebrafish? Can you please also mention the standard deviation of the same?
Yeah. So for zebrafish, which is interesting actually, because you would expect for small animal models that their heart rates will be quite high. For instance, in the mice, it's known that their heartbeats are much higher than humans. They're up to 600 beats per minute. In zebrafish, actually, the heart rates are much more similar to humans. So of course, it depends on if they are awake or anesthetized. So you have to be careful with this when you measure this. If they are awake, we see heart rates between, I would say, 150 to 200 beats per minute. If we do light trichain anesthesia, it will drop down to about 120, 140 beats per minute. That's about what we expect.
Thank you, sir.
Thank you, sir. Thank you for taking time from your really busy schedule. We are really grateful.
Thank you.
And
very
it
well.
was a lovely presentation, sir.
Thank you for the invitation.
Thank you. We have now come to the end of the webinar, and we have Ms. Elizabeth Reggie to propose the vote of thanks. Elizabeth, please.
Yes, thank you, ma'am. Good evening to all. On behalf of Department of Medical Biotechnology, I extend a very hearty vote of thanks to all our eminent speakers for sparing time from their busy schedule and gracing us with your talk and sharing with us your knowledge on zebrafish as an emerging model in research and its various future prospects. Thank you for making such excellent presentations and making this seminar interesting and meaningful. I'm immensely thankful to our director and HOD, Dr. Mansi Thakur, for always being there and supporting and encouraging us for organizing such a meaningful webinar. Finally, the wonderful participants who have joined us in this webinar, not only from our department, but also from other colleges. Thank you so much for your cooperation. I'm sure that all the participants who have attended today's webinar would use all of this gained knowledge into finding their way into research using zebrafish as their animal model. Last but not the least, I would like to thank the entire SBS team for their support and Mr. Anil for the IT support. Finally, I would request a few minutes from all of you to send us a feedback for the event. The link has been sent to your email, and the e-certificates for all the participants will be sent via mail by next week. Thank you everyone for joining us. Stay safe. Thank you.
Thank you so much, sir. Thank you. Thank you, everybody. And we can end this session now.